to be here. So today I will be talking about source coding for genomic information storage. This is joint work with Professor Olgica Milinkovic and Professor Zahi Weissman. So what do I mean by genomic information? I mean DNA, RNA, and proteins. And in terms of content, it doesn't only include nucleic acid and amino acid sequences, but also it includes information such as shapes of these molecules, probability scores, which tells you how confident you are with the measurements from the experiment, and the transcriptome information, and also protein abundance, and so on and so forth. And in recent years, there have been quite a few technologies that are developed to help us to obtain such information. For example, DNA sequencing, ChIP-seq, which helps us to know the interaction between DNA and proteins, RNA-seq, which tells us information about RNAs, and also RNA-shape that tells us the shape of the molecules. And also, there are quite a few initiatives that tries to obtain, store, and analyze such information. For example, the Human Genome Project, the 1000 Genome Project, ENCODE, TCGA, and so on. And in this talk, we will talk about some data we obtained from ENCODE. And in particular, we're interested in RNA-seq. This is a recently developed technology that is also called whole transcriptome shotgun sequencing. And it basically tells me what types of RNA strands do I see in a cell, and also how abundant do I see for each type of RNA. And in particular, this technology takes a snapshot of a cell. This is because that the RNA profile in a cell doesn't only change from organism to organism, but it also changes if you look at different time stages of the cell. So if I take a sequence of snapshots of the RNA-seq, then actually I get the time information of a cell as well. So RNA-seq is very useful if we think about biological discovery. For example, it can tell us what is the exact boundary between exon and introns. And also, from genomic medicine point of view, it can help us to do a lot of discoveries. For example, it has been demonstrated that you can distinguish transcripts between some oral cancer and normal tissues. As a result, this is a very important technology, and we're interested in the data that are generated from this. And in particular, we look at the type of data that is called expression values. And here is what it means. So suppose that you have some RNA strand, that is a red sequence here, that is existent in your cell. And it can be discovered by the RNA-seq. And having known the sequence, the ATGCs, the AUGCs, then you can map it according to the complementary nucleotides to the reference genome or the DNA strand, for example, for a human being. So you know the location that you can map it to the DNA strand, and then you can do it for all of the RNA strands in the cell. So now you get the whole information about RNAs, and now we are going to extract some numbers from this. So we call it expression values. So basically, I look at each nucleotide location, and I ask, how many times do I see an RNA that is mapped to this location? So for the first location, I don't see any RNAs, so I say the expression value is 0. And for the second location, I see 2 RNAs, so I say the expression value is 2. And then the next one, I say the value is 3, and so on. For some of the regions, there are no RNAs mapped to it at all, and this is the part that we call introns. And here, the expression value will be zeros. So now you get a whole sequence of integers that we call the expression sequence. And you can draw a sequence that would look like something like this. And this sequence tells me the information that how abundant a particular location at the DNA will be translated into an RNA. And this is very useful information, so we're going to process this type of data. And the expression value is stored in the so-called Wiggle format, or sometimes it's called WIG files. And this is the standard that is used in the ENCODE project. And it will look like something on the right. So this is a very small piece taken from a Wiggle file. So you have a sequence of nucleotide locations that are consecutive integers, and you have a sequence of expression values. So Wiggle format is developed to display arbitrary kind of dense and continuous data. For example, you can store in Wiggle format the GC% or probability scores and so on. As a result, our techniques that are developed in this work can be applied not only to RNA-seq but also to other applications. So this is a figure that I took from NIH website. You can see that in recent years, especially after 2007, the speed, the cost that you need to take in order to sequence a genome is decreasing. That is faster than the Moore's law. Or equivalently, I could say that the volume of genomic data is increasing at a speed much faster than Moore's law. And in particular, if we look at the ENCODE data set, 10% of all the files or 31% of all the total storage is dedicated for WIG files. As a result, from storage point of view, it would be very beneficial if we could compress such data, use source coding techniques. From communication point of view, here I will show you a little example I saw from New York Times. BGI is a company, a sequencing company based in China, and they produce equivalent to 2,000 human genomes per day. And if you send this kind of information through internet to the customers, it will take weeks. As a result, they had to turn to FedEx and ask the postman to physically deliver the hard disks to the door of their customers. So from communication point of view, if we want to deliver information electronically, then compression is not only beneficial, but also necessary. So this raises questions to information theory that trace back to Shannon. And in particular, we will focus on the source coding part. So you may have heard of some source coding algorithms such as JPEG, MPEG, and so on for multimedia. These kind of algorithms are called lossy in the sense that when you have some information and you pass it through an algorithm like this, you cannot recover the original file entirely. So in this work, we focus on lossless compression. So the compressed file and the original file are one-to-one mapped. Algorithms of lossless compression include Huffman coding, which is optimal only if you consider symbol-by-symbol compression. Also, Lampo-Ziv compression, which is universally optimal, but also has a constraint when only if you consider long enough sequences. And at last, arithmetic coding, which is near optimal algorithm, and it has only linear complexity in the length of the sequence. As a result, this will be the algorithm that we pick. And of course, source coding has been considered for genomic information storage, and there are quite a few works. Here we only list a few examples. For lossy compression, people have considered how do you compress a very large database into a tiny one so that can serve for similarity search. And for compression of whole genome sequences, the ATGCs, people have considered direct compression algorithms and also reference-based algorithms. The reference-based algorithm is based on the fact that the human beings have only 1% difference in their genomes. So you can take a reference genome and compress only the differences. However, not many work has been done for sequence of expression values. As a result, this will be our target. So let's come back to the file that we want to compress. And first, let's get a feel what it statistically looks like. So we can print out, plot out the empirical PMF of the expression values in the whole sequence. You can see that the alphabet size of the sequence is actually very large. You get up to 10 to the 6 or 10 to the 5 large expression values. And also the probability distribution more or less follows a power law distribution. And also, when you look at the larger integers, the appeared symbols are kind of sparse. And if we look more closely at this sequence, you can see that there are actually runs with identical expression values. As a result, we decide to first use a run length compression scheme. So what we do is, if I look at the first run, then I will record a run length of length 6, and then I will record the expression value of 22. For the second run, I will record the run length of 2 and value of 23, and so on. Now, suppose that I know the first location value, and suppose that I have the run length sequence and the value sequence, I can recover the original sequence. So this step is lossless. Run length compression has been used for many, many years, for example, in bitmap images and also fax machines. So now we can plot out the empirical PMF of the run length. So you can see that, again, it has very large symbol size, so the alphabet size, and also it's kind of sparse in the larger integer range. So if we look again at the derived sequences, we can actually see that adjacent numbers tend to have small gaps. As a result, we decide that we're going to use one more step in the processing of the sequence. We call it delta encoding. So basically, if I look at adjacent run lengths, I will take the difference of them and record it -4. And also, if I look at adjacent values, I will record the difference, which is 1. And you can write the whole sequence. So now, if I know the run length, and if I know the value, the first run length and the first value, and also the two sequences of differences, then I can recover the original sequence. So having these two different sequences, we're going to further compress it, and we're going to use arithmetic code. So delta encoding has been used not on symbol basis, but on file basis for HTTP file archiving and online backup. And also, it was used in the reference-based genome compression that I just mentioned before. So arithmetic code was developed in the 70s, and it basically tries to encode a whole sequence of symbols into a real value that is between the range of 0 and 1. And please bear with me if you're familiar with this algorithm, and I will explain it with a simple example. Suppose that we have a sequence with 3 possible symbols, A, B, and C, with probabilities 0.5, 0.33, and 0.17. So I will divide the range between 0 and 1 according to the probabilities. So I mark them with 3 different colors. So now suppose that the first symbol in my sequence is B. What I'm going to do is I'm going to use a real number in the green range. So any real number in the green range. So now if I see this dot, I know for sure that this value that I want to encode is B. Now, what I'm going to do next is I'm going to split the green region, again, according to the probabilities. And suppose that the next symbol I want to represent is C. Now I'm going to use a real number in this red region, and so on. So maybe next symbol I want to represent the A, then I will use a dot in the blue region. So now suppose a decoder knows the probabilities, and also the decoder knows this last dot in the blue region. Then it can trace back according to this graph and know that the sequence is B, C, and A. So basically, arithmetic coding is another kind of different radius when you try to represent a number. So arithmetic coding is proved to be only 2 bits away from entropy, which is the lower bound of a code length. And also, in practice, we can implement it with fixed precision algorithm. And also, as we said, it has only linear complexity. A variance of arithmetic code is range encoding, which operates on byte basis instead of bit basis. So it is faster in time. So here, notice in this graph that the probabilities is given beforehand. So in practice, we will take the frequencies that we see in the sequence of the 3 symbols. But also you can take arbitrary probabilities, and still the encoding and decoding will go through. So the probability, the choice of probability and the choice of this particular encoding algorithm are actually separate. So here are some simulation results that we made on the data from ENCODE. So you can see that we compare our work with some existing work. The first one is BigWig, the green algorithm. The BigWig was a standard in the ENCODE database, actually. So the second scheme we compared with is gzip, which is based on Lampo-Zip and Huffman, and it is used in Unix and Linux systems. And our algorithms, the range encoding and arithmetic code, is basically has demonstrates about 19 times improvement in terms of code rate. And one work that we didn't compare with is the recent published work called CWig, which also compresses the Wiggle files. And the CWig, according to their paper, they have about 3.5 times improvement in code rate. So far, we have talked about the basic algorithm that we used in order to compress expression values. But there are further considerations when we look at the data from application point of view or from implementation point of view. And I will talk about 3 of these considerations. Firstly, before we had used IID model, so we assume that all the symbols in the sequence are independent. But in practice, you can use more sophisticated models, such as case order Markov model, where every symbol in the sequence is depend on its prefix of length k. So in order for the encoding and decoding to go through, you actually have to maintain a table that contains all the possible k+1 strings. So if the alphabet size of the sequence is large, this table will be huge. It grows exponentially with k. And on the other hand, you could use some other techniques to shrink the size of the table. For example, you could use a dictionary that contains only the appeared k+1 strings. And this is the technique that is used in prediction by partial match. Another example of a model for generating the sequence is tree source model. Basically, every symbol in the sequence is dependent on a prefix of variant length. And this prefix can be found as a leaf in a given tree. This is a model that is used in content tree weighting. And also, for the above 2 models, you have to either store a dictionary or you have to store a tree. But alternatively, you can find a match in the past sequence for the current substring that you want to encode. And basically, you only need to have a pointer that points to something already happened and say, this part is going to be identical to what happened there. And this is the technique used in Lampo-Zip. Now, for a given sequence, we don't know what model it actually fits. So we can actually take all of these models and mix them together with different weights. And this technique is called context mixing. We have said that in arithmetic coding, the probability generation and the encoding are actually separate. As a result, once we have chosen a proper model and generates the proper probabilities, then we can feed it into the arithmetic coder. And in fact, we can also encode adaptively. As we go through the sequence, we update our knowledge about the probabilities. And for every step in the encoding, the probabilities could be different, as long as the decoder also is able to recover the same probabilities. And this will be the technique that we use, the context mixing. So we compare different algorithms, range encoding, arithmetic, and context mixing. We can see that range encoding has the largest code rate. Arithmetic has almost the same rate as empirical entropy, which is predicted by a lower bound. And then the context mixing can further reduce the code rate by about a quarter. However, we have to pay a price when we get this very low code rate, which is encoding or decoding time. We can see that range encoding and arithmetic code have more or less the same decoding time, but has much smaller decoding time, encoding time compared to the standard bigwig. However, context mixing takes a much longer time to encode and decode. And also something that I didn't show here is that this algorithm also requires a large amount of memory to implement. So the second consideration is random access. So a user looks at the whole expression value sequence, but this may not be all useful for the user. Instead, the user may ask for only a specific region in a specific chromosome. So if we compress the whole sequence as one file, then it is not possible to find where the requested substring you can find in the compressed sequence. So alternatively, we can encode the whole sequence into small blocks. So for every block, we have a separate encoding and decoding procedure. Now, if I'm looking for something in some of the blocks, then I can look for the corresponding compressed blocks and recover it. As a result, we can save some of the decoding time and also the space and memory and so on. Of course, we need to manage some of the auxiliary data. For example, we need to know where are the compressed blocks, because every compressed block has a variable length. And also, since we use the run length compression, so we only know the runs of the sequences. We don't know which locations they actually correspond to. As a result, if the user asks for a specific location, you have to be able to fastly retrieve which block it actually corresponds to. So here we compare that how much we gain if we use a larger block size in terms of code rate, but also how much we lose in terms of access time. So we can see that for different block sizes, compared to arithmetic coding, to encode the whole sequence, we lose at most about 10% in code rate. And in terms of access time, so far we don't see much difference when we choose different block sizes. However, this may be due to the fact that the query size we chose is kind of too large compared to the block size. So since this is still an ongoing project, our prediction is that if we make the query size much smaller, then smaller block size will benefit us. And at last, the last consideration would be distributed computation. So we have this very large database in the medicine institute, and they have very high power in computation. However, right now our algorithm only uses one processor to do encoding and decoding, and it will take several maybe 100 seconds to finish this. As a result, we say why not use quite a few of the servers in the data center. So here is a very simple idea. So I take a whole Wiggle file that contains all the expression values of all the 24 chromosomes, and I send it to a few of the servers. And every server only processes part of the chromosomes, and then they can combine their information together, either compress or decompress. So we tested this idea in a single computer, but with multiple processors. And we can see that if we increase the number of processors, the encoding time decreases. But it also kind of has a floor kind of effect. This is due to the fact that there is some overhead when we do distributed computation. For example, you need some time to do memory access, and also all the processors need to talk to each other. And also, we divide the file according to the chromosomes, but different chromosomes have different lengths. For example, chromosome 1 in human genome is the longest in all of them, and it is actually much, much longer than chromosome X and so on. So in fact, the longest chromosome will be the bottleneck of this system. It will affect the delay of how we encode and decode. So in summary, I hope that I have convinced you that genomic information cries out for compression. And we have introduced a specific algorithm for lossless compression. And also, we have talked about different considerations in terms of implementation. So there are quite a few open problems in terms of RNA-seq. For example, we could try to use some lossy compression algorithm, because due to experimental errors, you could see that even though the a region corresponds to the same exon, we should expect to see the same expression value. But due to the errors in the experiment, we see different numbers. So therefore, if lossy compression is applied, maybe we can actually decrease the noise. And also, it would be very nice to be able to build a database for transcriptome. This can be used for biological purposes, but also from compression purpose, compression point of view, it will help us to further reduce the size of the files. And at last, it would be nice to be able to classify different tissues, for example, tissues from different organisms, or tissues for cancer cells or healthy cells. So I would like to thank Professor Olgita Milinkovic and Professor Zaki Weissman for very helpful discussions and guidance, and also Center for Science of Information for very general support. And I have one last word to share with you. So recently I watched this movie, spoiler alert, that the main character was able to access 100% of her brain, which our normal person only access 10%. So she accumulates this huge amount of knowledge, and she has very strong abilities, but she doesn't know what to do with it. So she turned to this neuroscience professor from MIT, who is played, of course, by Morgan Freeman. And then he said, if you're asking me what to do with all this knowledge you're accumulating, I say pass it on. So I feel that I'm very lucky to be able to be here at the summer school to witness the part that people are passing on the knowledge. So thank you very much.